Good afternoon, everyone. My name is Nicholas Horton, and it's my honor and privilege as chair of the Committee of Presidents of Statistical Societies to welcome you to this year's COPSS Awards and Fisher Lecture. COPSS is an organization that represents the original entities that formed the Joint Statistics Meetings, the American Statistical Association, the Eastern and North American regions of the International Biometric Society, the Institute for Mathematical Statistics, and most recently, the Statistical Society of Canada. COPSS is more than 50 years old and works to improve inter-society communication and cooperation, and also sponsors a number of high-profile awards, a number of which we'll be awarding today. COPSS consists of the past presidents, and presidents-elect of the 5 member societies, as well as a chair and my able secretary and treasurer, Wendy Liu of the University of Toronto. COPSS meets regularly with the Friends of COPSS, listed here, as a way of broadening these discussions of important issues facing the statistics profession. Just a reminder and a shameless plug for the COPSS book, edited by Shihong Lin, entitled Past, Present, and Future of Statistical Science, which is available at bookstores or for free download from the COPSS website. Newly available this year from CRC Press and also fine bookstores is Amanda Golbeck, the recipient of last year's Elizabeth Scott Award, entitled Equivalence Elizabeth L. Scott at Berkeley. The COPSS Awards presented today represent the work of some of the finest work we have in the statistics profession. We will be awarding today the Florence Nightingale David Award, the George W. Snedeker Award, the President's Award, and then the Fisher Award, and then the lecture that's associated with that award. Other COPSS awards presented in even years are the Elizabeth Scott Award for an individual who has helped foster opportunities in statistics for women. So to ensure that the best work in statistics is rewarded appropriately, it's very important that you submit nominations for these COPSS awards. I would encourage you to think about possible nominees so that their work can be recognized and really acknowledged. So coming up for next year, we will be awarding the President's Award, the E.L. Scott Award, the Fisher Lecturer, and the nominations for the first 2 of those are due on January 15th. The Fisher Lecturer and Award nominations are due on December 15th. So before we begin, I want to also acknowledge the work of the COPSS Awards Committee that was ably chaired this year by John Brewster of the Statistical Society of Canada, and really represented a lot of efforts to ensure that the awards process is fair and the awards of the highest quality. And now it's my great pleasure to be introducing Roxy Peck, who will be announcing the recipient of this year's Florence Nightingale David Award. Thank you, Roxy. Come.
All right. Well, thank you very much. It's an honor to be here to present this award. This award honors a female statistician who serves as a role model to other women by her contributions to the profession through excellence in research, leadership of collaboratory groups, statistics education, and service to the professional societies. A little bit about Florence Nightingale David. She was an accomplished statistician in combinatorial and probability theory, and an author and editor of almost too many books to mention. The one that's mentioned here, I think, is considered a classic now. It was a book called Games, Gods, and Gambling on the History of Probability Theory. She was the first chair of the Department of Statistics at the University of California, and also the first recipient of the Elizabeth Scott Award. This particular award is important to me because my very first class in statistics was a course from Dr. David out of the Games, Gods, and Gambling textbook, and it was what kind of piqued my interest in studying statistics. And later, I joined the graduate program at the University of California at Riverside. So it's a real pleasure and honor for me to be able to present this particular award. I want to thank the members of the awards committee that you see here. We got a large number of just outstanding applications and nominations this year, and these people worked very tirelessly going through the nomination packages and working on the selection of this year's winner. So I thank them. And now your suspense, I'm sure, is peaked, so we'll announce the 2017 winner. And the 2017 winner is Zhihong Li. She's the Henry Pickering Professor and Chair of the Department of Biostatistics at the Harvard School of Public Health. I get to tell you a little bit about Zhihong before we give her a chance to say a few words. I want you to think back to the early slide that described this award that says that it honors somebody who excels in research, leadership, service, mentoring. Zhihong really excels in all of these fields. It was kind of impressive because you usually think about, well, which of these areas does this particular person, really stand out in, and she just stood out in every single one of these. In terms of research, she's recognized for her collaborative research in genetics, public health, and medicine, and considered a leader in that field. And she has over 240 papers published in both top statistics journals and also in applied journals like Nature and the Journal of the American Medical Association. It's quite an impressive list, and you also saw her name mentioned as the editor of one of the COPSS books that's available now. Honors and awards, she's received many. Nick only gave us one slide for each of these topics, so this is just a few of the honors and awards that she's received, including the COPSS President's Award in 2006. She's a fellow of the ASA, IMS, and ISI, and as it says at the bottom, and many more. But what impressed me, I think, the most about Dr. Lin's nomination was how the letter writers described her as a person of passion and generosity, especially in mentoring and nurturing students and early career statisticians. They spoke very enthusiastically and described in great detail the time and the concern that she puts into that effort. She's also served in leadership positions, as I mentioned before, the chair of the Harvard Biostatistics Department. She was actually the COPSS chair back in 2010 through 2012, and has done editorial service as well. So that's about as much as I can tell you in 2 minutes, but I would like to tell you that there's much more, and I would encourage you to take a look at Dr. Lin's work and service. I'll just read the citation before inviting her to come up and say a few words. The citation that our committee wrote is to recognize her for leadership and collaborative research in statistical genetics and bioinformatics, and for passion and dedication in mentoring students and young statisticians. So, Zhihong, I'd like you to invite you up here to say a few words.
Back over to there. Congratulations.
I'm very humbled and honored to be receiving this award, and I would like to express my sincere appreciation to the COPSS Davis Awards Committee for recognizing me with this award. So this moment is much bigger than me. It is for women in statistics and all of you who have made endless effort to support and promote diversity in our profession. Being able to contribute to statistical profession has been a joy and a privilege that I'm grateful for and cherish every day. Statistical science challenges and inspires me. I cannot imagine a world without it. I cannot imagine doing anything more rewarding. I have been very lucky to have had great caring mentors who have supported and inspired me throughout my career, such as Norm Breslow and Rick Harrell, Louise Ryan, Rod Little, Jim Ware, and Marvin Zallen, and many colleagues and friends, and also many talented students and postdocs I have worked with. If I have done anything right, it is because they showed me how, and I basically try to do the same for students, postdocs, and junior statisticians. I would like to particularly encourage junior statisticians, especially young female students, to pursue your passion and dream. Someday, it will lead you to a stage like this. I would like to congratulate all women statisticians for your contribution to statistical science. I would like to thank the statistical community, COPSS, the 5 charter societies of COPSS, and friend societies for having done an amazing job in supporting and promoting women, and also for all of you who have helped advance this mission. Thank you.
Wonderful. So now it's my honor to be introducing Paul Rathouse, who will be announcing the winner of this year's George W. Snedeker Award.
Thank you, Nicholas. The George Snedeker, good afternoon, everyone. The George Snedeker Award honors an individual who's been instrumental in the development of statistical theory and biometry, and also has had a noteworthy publication in biometry within the past 3 years. Dr. Snedeker, as some of you may know, was a pioneer in improving the quality of scientific methods concerning statistical methodology, and was the first director of the statistical laboratory at Iowa State starting in 1933. He had a famous textbook and worked in the areas of experimental design, sampling, and he was instrumental in the worldwide use of statistical methods, both in applications and in theory. The awards committee is listed here, and I want to thank all of them. They worked very hard. We had an exceptional number of very highly qualified nominations this year, and they pursued their charge very diligently, so I'd like to thank them. And without further ado, I'd like to announce the winner of this year's award, Professor Aurore de l'Aigle, who is Professor of Statistics at the School of Mathematics and Statistics at the University of Melbourne in Australia. Professor de l'Aigle has many honors, and a few of them are listed here, and I'll just mention one of her most recent ones is that she's a fellow of the Institute of Mathematical Statistics. She's received a variety of other fellowships and awards, and like I said, I've just named a few here. She's also served in many leadership positions, including serving as associate editor for more journals than I can believe, which is quite impressive. And then she's also an associate chair of the current meeting, so we'd like to thank her for that. And she just finished her role as executive secretary of the IMS yesterday. So that's the updated information here. We thank her for her service in those roles. Her research, her main area of methodology, she's covered a variety of areas, but her main area of methodology has been in the area of parametric and non-parametric methods for group or pooled testing of biological samples. These methods are used to assess the presence of a biological contaminant through a test, for example, a disease in the blood of individuals, and in particular they address group testing, which deals with a situation where data from patients or individuals are pooled prior to testing, sometimes with and sometimes without covariates. She's worked on both parametric and non-parametric methods to estimate prevalence from this kind of data. Non-parametric methods, of course, are more robust against model failure, but they require a considerable level of special theory and attention to tuning parameters. Aurora's work has been instrumental in developing this body of methodology through a series of papers that have advanced important applications to estimate prevalence either crudely or as a function of key covariates. And she's worked on complex extensions, in particular improved estimators through homogeneous grouping, multivariate variations, and aggregated covariates. The paper for which the award is given is entitled Non-Parametric Methods for Group Testing Data Taking Dilution into Account, with her as lead author and co-author, the late Professor Peter Hall. This paper addresses bias that can result from misclassification of the test result, and in particular deals with the fact that that bias can arise from variation in group size in the pooling, and that particular issue is especially challenging to handle. The official citation is for fundamental and groundbreaking contributions to the statistical theory of group testing of pooled laboratory samples and for contributions to measurement error methods and density estimation. Congratulations, Aurora.
Thanks very much, Paul, for these nice words, and thanks very much to the committee for awarding me this prize. It's a real honor for me to receive this award. I want to thank my long-term collaborator, Alexander Meister, with whom I wrote my first paper on group testing problems during one of his visits to Melbourne. But I want to dedicate this award to my collaborator, mentor, and friend Peter Hall, with whom I wrote the Biometrica paper that was just cited, and who passed away last year in January. Peter was an extraordinary, kind, gentle, unassuming, and generous person, of the type that most people do not even have the chance to meet once in their lifetime. In addition to his amazing mathematical skills, what was fantastic about him was his passion and enthusiasm. He got immediately interested when I talked to him about the group testing problem, and we wrote a sequence of papers on the topic. Working with him for 12 years and being his office neighbors for 10 years will forever be one of the most amazing experiences in my life. Thank you, Peter.
Now I'd like to invite Amy Herring to the stage to present this year's President's Award.
Thanks very much. The President's Award is presented annually to a young member of one of the 5 participating societies of COPSS. This award is presented in recognition of outstanding contributions to the statistics profession and is widely regarded as the preeminent award in the field of statistics. The award criteria involve contributions to the development or application of statistics as a discipline, comprising either a single contribution of extraordinary merit or an outstanding aggregate of contributions to the profession of statistics. This award has age criteria. It's typically granted to an individual who has not yet reached his or her 41st birthday during the calendar year of the award. However, in the special case of an individual who has received a terminal degree in statistics less than 12 years prior to the nomination deadline, a nominee will remain eligible until the 46th birthday. As you can see, COPSS has a formula for putting together committees, which is one member of each of the participating COPSS societies, as well as a member at large of COPSS and a former winner. This year's committee worked very hard, and I'd particularly like to thank Peter McCullough, who couldn't be here today. We had a lot of fun reading the amazing applications put before us, and we'd also like to thank the nominators who took the time to put together these testaments to the work of those who were nominated. And the 2017 President's Award winner is Tyler Vanderweel, Professor of Epidemiology. Tyler is professor in the Department of Epidemiology and the Department of Biostatistics at the Harvard T.H. Chan School of Public Health. Tyler is very much a polymath, a Renaissance man. He received his bachelor's and master's degrees in mathematics, philosophy, and theology at St. John's College in Oxford, master's degree in economics and applied finance at University of Pennsylvania, master's and PhD in biostatistics at Harvard University. From there, he went to the University of Chicago, Department of Health Studies, and is now professor at Harvard. He's done foundational work in methods for delineating causal mechanisms with many peer-reviewed, high-impact papers in statistics, in the epidemiologic literature, and in subject matter areas. He also has a new book. He's won many awards for his work, including from the American Statistical Association, the Causality in Statistics Education Award, the American Public Health Association's Mortimer Spiegelman Award for Outstanding Public Health Statistician. He's given many distinguished lectures. He is a founding editor of the journal Epidemiologic Methods and serves as associate editor or on the editorial board of many journals that are perceived as the top in their fields, including the Annals of Statistics, the American Journal of Epidemiology, and Epidemiology. Tyler Vanderweel is awarded the President's Award for fundamental contributions to causal inference and the understanding of causal mechanisms, for profound advancement of epidemiologic theory and methods, and the application of statistics throughout the medical and social sciences, and for excellent service to the profession, including contributions to teaching, mentoring, and bridging many academic disciplines with statistics. Congratulations, Tyler.
Thank you very much, Amy and Nick. It is a great honor and truly humbling to receive the COPSS President's Award today. As one looks over the list of past recipients, one cannot but feel a sense of duty to try to live up to the contributions made by them, not simply those prior to the award, but those contributions that followed it. If I'm able to continue to contribute to the profession of statistics in some small way and to try to live up to the contributions of these others, I will be grateful indeed. I owe a great deal of thanks to a great number of people. I would like to thank the awards committee for the great honor of being here today and for considering my contributions, which have sometimes been more conceptual and often focused on the needs of the biomedical and social sciences, as being of value to the profession of statistics, as indeed I believe they are. It is from the foundation provided by statistics that so many other disciplines are able to proceed. I would like to thank those who nominated me, Ji-Hung Lin, and those who wrote letters on my behalf for their encouragement, mentorship, and intellectual collegiality. I would like to thank my parents for their support, both in life generally and in my education and intellectual development. I would like to thank my wife, Lisa, and son, Jonathan, for their love, for their unending support, without which I could not continue, and for putting up with numerous late nights and occasional late mornings in my various, sometimes abstract, scholarly pursuits. I would like to thank the numerous extraordinary teachers I have had over the years for excellent training in mathematics and statistics, but also for continually sharpening my ability to reason and to think clearly about concepts. I would like to thank my colleagues at Harvard and in causal inference, and in particular Eric Chekin and Miguel Hernan, from whom I have learned a great deal and who have always been a joy to work with. And finally, a special debt of thanks to my dissertation advisor, Jamie Robbins, whose mentorship from graduate school onwards has constantly challenged me to think clearly and deeply about ideas and concepts and to focus on what appears most central and important. It has been a privilege working with him these past years, and a privilege also to work with my colleagues throughout Harvard, throughout causal inference, and within statistics and epidemiology broadly. Very little can be achieved without a community, and it has truly been an honor to have such wonderful, brilliant, and supportive people around me, and to be a part also of this broader community of statistics. Thank you.
So now, last but not least, we look forward to inviting Kiros Borhani to the stage to be introducing this year's recipient of the Fisher Award and lecture.
On behalf of the COPSS and this year's selection committee, it's my distinct pleasure to present the winner of the 2017 Fisher Award and lecture. The R.A. Fisher Lectureship was established in 1963 by COPSS to pay tribute to the contributions of Sir Ronald A. Fisher, to honor the work of a present-day statistician, and the award recognizes meritorious achievement and scholarship in statistical science and highly significant impact of statistical methods on scientific investigations. So this year's committee was comprised from COPSS and also the 5 participating statistical societies, as well as the past awardee, Professor Jeff Wu. The committee had a very difficult task of choosing a winner from several outstanding and well-deserving candidates. So I would like to take this moment to thank the committee members for their careful and thoughtful deliberations throughout the selection process. So it's time now to announce this year's winner of the Fisher Award and lecture, Professor Robert COPSS. So Professor COPSS is a Maurice Fogg Professor of Statistics and Computational Neuroscience. He is in the Department of Statistics and Machine Learning Department. He's also the interim co-director for the Center for the Neural Basis of Cognition at the Carnegie Mellon University. Professor COPSS received his PhD in statistics from the University of Chicago in 1980. He's a renowned statistician whose work has spanned a wide range of accomplishments through his groundbreaking foundational work on statistical methodology and theory and deep thinking in and substantial contributions to application areas, in the process shaping how scientists think about fundamental problems in their own fields, and most notably in the area of neurosciences. Professor COPSS has received numerous prestigious awards throughout his career in recognition of his seminal contributions to statistics, both in foundational work in statistical theory and their applications to scientific investigations, again, particularly in the area of neuroscience. So he has received numerous awards, but just to give a partial list, the Leonard Savage Award for Outstanding Thesis in Bayesian Statistics and Econometrics in 1981. He was named Fellow of ASA in 1990. He was named Fellow of IMS in 1991. He was named Fellow of the American Association for the Advancement of Science in 2002. He gave an IMS Medallion lecture in 2002. He was also the INR Presidential Invited Lecturer in 2010, and he received the ASA Outstanding Applications Award in 2013, to name a few. So the citations that the committee prepared for him goes as follows. For groundbreaking contributions to several areas of statistics, including use of differential geometry in statistical theory, as well as theory and methodology of Bayesian inference. For strong commitment to the application of principled statistical thinking and modeling to problems in computational neuroscience, and for his strong dedication to training of students and users of statistics. Congratulations.
Thank you, Kiros and Nick. Good afternoon, everyone. Let me begin by thanking the whole of the COPSS Fisher Lecture Selection Committee, as well as the people who nominated me and supported my nomination. It takes all kinds of people to contribute to the field of statistics, and I'm just glad to be one of those kinds. I also want right away to thank the many knowledgeable, talented people I've worked with over the years, including those I've worked with in the brain sciences at Carnegie Mellon and the University of Pittsburgh, most of whom are listed on my website. The lessons learned because of those collaborations about the role of statistics in science are the subject of my talk. It's a huge honor and a great pleasure to be delivering this year's R.A. Fisher Lecture. I remember first coming to the joint statistical meetings more than 30 years ago and sharing with some of my contemporaries my eagerness to hear the keynote address of one of the stalwart senior statisticians. But honestly, never in my wildest imagination could I have visualized myself someday being that old. And yet, here I am. In fact, next month I'm going to be 65, so I'm conscious of being a senior citizen, and while I've adjusted very comfortably to often being the oldest person in the room, it has its challenges. As a teenager, I was already an absent-minded professor, and at my advancing age, this has become downright dangerous. Not too long ago, I was at an off-campus meeting and was walking back to my car, deep in thought, got in and had a moment of panic because the steering wheel was gone, and I thought, who would steal a steering wheel? And then I realized I'd gotten in the back seat. I sincerely wish I were kidding. A different kind of challenge comes in the form of numerous unwanted age-related messages. The other day, while I was waiting in line for some fast food, I pulled out my phone and started reading the New York Times when suddenly I was interrupted by a pop-up displaying an equation. My first thought was, that's a stupid equation. Then I recognized it was a puzzle. And even though when I was a kid I loved math puzzles like this, now I find them just plain annoying. I don't want anything to do with it. And worse than that, I got kind of pissed off that the software the New York Times is using thinks that I'm so old that I need this kind of puzzle to keep my brain functioning properly. And as I thought about it more, I got even more pissed off at the software because it was smart enough to know how old I am, but not smart enough to know that I work with equations for a living. And furthermore, the equations I study are actually consequential, unlike that irrelevant one they just showed me. But I took a deep breath, and I said to myself, okay, it's a puzzle. The answer is probably an integer. There are square root signs, so it's probably a square. 15 is odd. The difference of two squares is odd. 49 plus 15 is 64. The square root of 49 is 7. The square root of 64 is 8. 7 plus 8 is 15. Done! I did it! Here's the thing. This made me so happy, it's embarrassing. I had such a feeling of accomplishment that I went home and took a nap. This is all a way of saying that while my technical abilities may be diminished, I still like to tell stories. And the story I've put together for you today is one I hope you'll find pretty interesting. It will end up connecting rather well with Fisher and might even give you a new way of looking at Fisher's contributions and his perspectives on our field. The brain sciences seek to understand how neural activity produces behavior. Many people find the subject fascinating, partly because our brains define us, but also because they may be viewed as information processors, which opens the door to quantitative analysis. Anyone who ventures into the subject is struck, on the one hand, by the brain's daunting complexity, yet, on the other hand, by the remarkable and inspiring progress made in the brain sciences, especially over the past 50 years. There's also the obvious social need for further advances in the brain sciences. Everyone in this room has a family member or a friend or a family member of a friend who's been touched by some dysfunction of the brain, including, of course, psychiatric and neurological disorders and a variety of other disorders, including addiction, which I'll mention again later. For all of these sources of human suffering, there are many treatments and substantial knowledge, but for none is there any detailed understanding nor a satisfactory cure. This was the motivation behind the current BRAIN initiative, which is a very helpful effort to fund the basic science needed to understand brain function and dysfunction, but from my perspective, it is remarkably underserved by statistics. This is the primary message of my lecture. I'm going to deliver this message by showing you a few examples of scientific questions that have generated interesting statistical problems, by reviewing for you at a high level the statistical ideas used to solve those problems, and then by observing that such solutions require an appreciation of the statistical paradigm, which is the name I'm using for what I understand to be the essentially universal guiding philosophy promoted by advanced statistical training. Having described the importance of the statistical paradigm for the brain sciences, I'll then say a few words about the importance of the statistical paradigm more broadly. Excuse me. Before I get going, I want to suggest to those who want to learn more a review paper I wrote with 24 other authors, about a third of whom I would call statistical, the rest being biophysicists who model neurons and neural networks. And there's also this book, Analysis of Neural Data, from which I'll be taking several figures today, labeling them KEB for Cass Eden Brown. Emery Brown is an MD PhD that is a practicing anesthesiologist with a PhD in statistics, who is a professor at both Harvard Medical School and MIT, and he's also a renowned neuroscientist from whom I've learned a great deal and collaborated with since I first got into this field 19 years ago. Uri Eden was Emery's first PhD student, and he's now a tenured faculty member in the Department of Mathematics and Statistics at Boston University. Most people outside of neuroscience are exposed to it mainly through fMRI studies, which often show pictures of brain activity under particular experimental conditions. But my own research has focused mostly on analysis of neural spike train data, which is the kind of data that appear in the examples I'll go through today. So I need to give you some background. And for this, I always begin with a striking graphic that does a very nice job of illustrating something I think most of you already know, which is that the brain is located in the head. And you probably also remember that neurons communicate primarily through rapid electrical discharges known as action potentials, or spikes, which take place over approximately 1 millisecond. When placed on an axis representing the several hundred milliseconds it takes to execute a behavior, such as moving your eyes towards something that captures your attention, the neuron will emit multiple spikes, collectively known as a spike train. A spike train consists of an irregular sequence of points in time, and in probability and statistics, an irregular sequence of points in time is modeled as a point process. I'll come back to that shortly. I need to add that action potentials are generated as a result of inputs from other neurons across synapses. And these inputs can be either excitatory or inhibitory. Excitatory inhibits increase the electrical potential, that is the voltage, while inhibitory inputs decrease the voltage, with excitatory or inhibitory inputs driving the voltage toward or away from the threshold at which the neuron will fire. You can see this in the recording. In this recording, you can see in this recording how the subthreshold voltage meanders up and down due to the excitatory and inhibitory inputs. The subthreshold voltage is often modeled as a random walk of upward and downward voltage steps drifting toward the firing threshold. And for theoretical purposes, after passing to the limit, the random walk becomes Brownian motion with a drift. So here's a simulation made by a graduate student in statistics and computational neuroscience at Carnegie Mellon, Spencer Kerner, showing above some of the excitatory and inhibitory inputs in blue and red as time evolves along the x-axis, and below the resulting voltage together with the output spikes, which roughly resemble real data. I'm playing the simulation about 10 times more slowly than it would occur for a real neuron. This random walk summation of inputs remains one of the standard conceptions of neural behavior. Now, a fundamental question in neurophysiology is, how do neurons carry information? And the simplest textbook answer is that a neuron responds to a stimulus by increasing its firing rate. Here's another textbook drawing showing a monkey trained to look at the center of a screen in front of him while an electrode records the spikes from a single neuron in his primary visual cortex, also called area V1, where information from the eye first enters the cortex, the gray matter of the brain, which is so highly developed in humans and in non-human primates. On the left is a picture of a typical electrode sitting near a neuron from which the electrode records activity, and on the right in the drawing you see that when a bar of light is moved across a small segment of the visual field known as that neuron's receptive field, it starts to fire much more rapidly than it would fire when the bar shines somewhere else in the visual field. By the way, the recording procedure is painless. We vertebrate animals have no ability to feel anything inside our brains. When this kind of response of neurons in primary visual cortex was first observed, in what became a very famous series of experiments, many of these neurons were also observed to respond preferentially to a particular orientation of the bar of light, as shown here on the left in part A, with a firing rate of a particular neuron in spikes per second, or hertz, on the right in plot B, plotted against the orientation angle measured clockwise from the orientation that produced the highest firing rate. This is called orientation tuning. And the idea that neurons are tuned to particular features of a stimulus is now ubiquitous in neurophysiology. Thus, much neurophysiology and much statistics is aimed at figuring out how neural firing rate is related to behaviorally relevant variables. But what about variability? Especially in the cortex, spike trains are highly irregular. In the figure on the right, each dot represents the mean firing rate across many repetitions of the experiment, that is, many repeated trials. But you'll notice the Poisson-like tendency for the variability to increase with the mean. This is also extremely common. And when we look in more detail at spike trains across many repeated trials, shown in the top plot, we again see variability both in the spacing of the spikes within trials and in the spike times across trials. In the bottom plot is a histogram made by counting the number of spikes within time bins across all the trials, then normalizing in units of spikes per second. The histogram shows the way the firing rate evolves over time, but it has done that by averaging away the trial-specific variability. Those are some basic characteristics of neural firing patterns in a single neuron recorded from a single electrode. These days, recordings are also made simultaneously from multiple electrode arrays, and new electrode arrays with ever greater numbers of recording sites are being developed by many neuroengineering labs and companies. One popular electrode array, known as a Utah array because it was developed at the University of Utah, is shown here next to a US penny. It has 96 electrodes in a 10 by 10 grid with the corners removed, and it can record anywhere from about 25 to more than 100 neurons simultaneously. I'm going to play a short video displaying Utah array data collected by former Carnegie Mellon student Ryan Kelly, now at Google, and Matt Smith, now a professor at the University of Pittsburgh. The electrode array was implanted in primary visual cortex, area V1, of a monkey, as in the cartoon I showed you earlier, except now you'll see real data recorded with the Utah array when the monkey was watching a movie, which will play at the top part of the figure. The circles are receptive fields of individual neurons, which turn red when the neuron is firing rapidly. Down below are the spike trains, from 128 neurons recorded simultaneously. This illustrates a typical statistical challenge of figuring out what these spike trains are responding to in the visual image. Okay, that was the background material. Now I'm ready to describe my selected examples of neuroscience questions that have led to interesting statistical problems and solutions based on the statistical paradigm. Out of dozens, I've chosen 3. I begin with the question, how are memories consolidated? The work I'm going to tell you about, very briefly, is centered on the hippocampus, a region of the brain that lies below the cortex and was first shown to be essential to memory storage by Brenda Milner and her colleagues in Montreal after a patient known as HM had his hippocampus removed during surgery for intractable epileptic seizures. The surgery succeeded in greatly relieving him from seizures, but left him unable to store new long-term memories for people, places, and events. These studies led to many others, and it became clear that long-term memories can become much stronger over the hours or days following initial memory storage, a process known as consolidation, presumably as neurons in the hippocampus send signals to and strengthen connections with other brain areas, which is thought to occur in a process of replay of firing patterns in the hippocampus. This may be one of the benefits of sleep or of non-sleeping rest. Here are two representations of data from a single neuron in the hippocampus of a rat while foraging in a circular pen approximately 2 feet in diameter after chocolate pellets had been sprinkled throughout the pen. On the left, in blue, you see the path of the rat, and the red dots are the locations of the rat when this one neuron fired. You can see the tendency for the neuron to fire whenever the rat ran through a particular location. And on the right is a visualization of a statistical model of the so-called place field for that neuron. The place field being analogous to receptive fields I showed you previously. Rodents have been used extensively to describe hippocampus activity in detail during such navigational memory tasks, including the phenomenon of replay of spiking patterns. This slide from my colleague Uri Eden illustrates spiking patterns among 13 neurons recorded simultaneously from a rat's hippocampus while the rat runs down a linear track. And you can see, with the help of the color coding, that the neurons have been ordered according to their place fields. On the left, you see the replay activity of those neurons when the rat was at rest. But here, there's not an exact match of the pattern. And in fact, each time the rat runs down the track or rests, a somewhat different pattern occurs. So to investigate such phenomena, how should we define replay? A reasonably straightforward strategy is to fit statistical models for the place fields and then use them to assign probabilities to particular patterns. This strategy, the one Uri followed, is based on point process regression models. It is described in some detail in our book, and it has roots in work of David Brillinger, which he described in his Fisher lecture now almost 30 years ago. The idea is conceptually very simple. We can consider irregular series of time points in continuous time to be a point process, or we can consider small time bins, such as 1 millisecond, the approximate width of an action potential, and create a corresponding binary time series with ones in bins where a spike occurs and zeros in bins where a spike does not occur. And let me mention that this picture is inaccurate because with our usual time resolution of 1 millisecond time bins, the ones are relatively rare, so we might expect to, say 1 spike in every 50 time bins. For a Poisson process, these continuous and discrete representations are related by saying that the probability of a spike within a particular time bin, here t through t plus delta, is approximately lambda times delta, where lambda is the intensity, which we can allow to vary as a function of time. The intensity is the theoretical representation of the firing rate. And as soon as the intensity is specified, we get the joint probability density function, the PDF, of the spike train, written in terms of the intensity. This, in turn, gives us a likelihood function so that we can fit the Poisson process model to the spike train data, which are counts of 0 or 1, using Poisson regression. That is, we can use standard software for generalized linear models. It's very straightforward. What makes Poisson processes special is that the intensity does not depend on previous spike times, and in this sense, the process is memoryless. Parenthetically, let me pause to try to avoid a potential confusion. The data are binary, so you might be expecting logistic regression instead of Poisson regression. However, because the spikes are rare, the probability of a spike in any particular time bin is very small, and therefore, in this context, Poisson regression and logistic regression are essentially equivalent. Now, I was saying that the Poisson processes are special because they are memoryless. The remarkable thing, which David Brillinger observed, is that the generalized linear model methodology extends immediately to general point processes simply by introducing a covariate vector, written here as xt, which may include past spike times. This is important because neural spike trains are found empirically to deviate somewhat from Poisson processes, as they should, theoretically, based on that random walk and Brownian motion conception I showed you earlier. The notation that I'm using, xt, I use as a way of emphasizing that we can condition on any variables we think might affect the neuron's firing rate. So xt might not only include past spike times, making the point process non-Poisson. It could also include features of a stimulus, or the animal's location, or inputs from other neurons, or aspects of the experimental design. In some of our applications, xt is low-dimensional, and in some it is high-dimensional. But importantly, just as we can use generalized linear modeling methods to fit spike train data with a Poisson process, so too can we use generalized linear modeling methods to fit spike train data with a more general point process. This is what I like to call point process regression, with the understanding that by modeling the intensity function, we can bring to bear all of our modern regression technology, typically combining Poisson regression with non-parametric function fitting and often penalized regression. That's how the place field on the right was found, and it's also how replay can be analyzed from a rigorous statistical perspective. Although it might take considerable work, it is conceptually straightforward modeling within the statistical paradigm. The second of my neurophysiology questions is this: how does synchronous spiking activity of neurons, possibly due to oscillations, contribute to information transmission? This is a question that's been posed and debated in the literature quite a bit. Two neurons are typically said to fire synchronously when they fire nearly at the same time, as illustrated here for time bins of width 5 milliseconds. The red arrow indicates a 5 millisecond time bin during which both neurons fired. This question about synchrony has been raised because the input voltages at the synapses decrease rapidly, roughly exponentially, as illustrated here, so that two or more inputs will have a stronger cumulative effect if they arrive at nearly the same time. Furthermore, synchronous spiking could result from network oscillations, while oscillations in many parts of the brain have been observed under various circumstances. To make sure you know what I'm talking about, I'm going to show you a dramatic visualization of brain network oscillations. This comes from electroencephalogram, EEG data, taken off electrodes on the scalp from a patient of Emory Brown's during anesthesia while undergoing surgery. The figure includes both a snippet of the raw time series on top and a spectrogram based on the Fourier transform across moving time windows on the bottom. The y-axis of the spectrogram is frequency in hertz, the x-axis is time in seconds, and the color indicates power, with red corresponding to high power and blue low power. In statistical terms, the spectrogram is telling us how much variation in the raw signal is associated with each frequency. Power is variance, so the spectrogram displays the components of variance due to each frequency. Here, the spectrogram shows high power, that is high variance, due to a pair of frequency bands, one roughly around 1 hertz and the other around 10 hertz. Both of these strong oscillations can also be seen in the raw signal above. Emory's work has emphasized that these very powerful oscillations are fundamental to the effectiveness of anesthetics in removing consciousness. But, as I said, oscillations have been observed under various conditions and have been associated with many brain functions, often at higher frequencies. This has led to considerable discussion of the potential computational purpose of oscillations, and a leading idea supported by interesting circumstantial evidence is that communication across brain areas could be facilitated by oscillatory activity. For example, different brain areas must communicate when you pay attention to something in your visual field. Somehow, the brain selects that thing being attended to, and multiple brain areas are engaged in processing the information it provides. Researchers have pointed out that an oscillation can drive many neurons to fire together, producing neural synchrony, which could lead to increased activity in relevant downstream neurons, thereby being an important source of information transmission. It's not hard to demonstrate this kind of thing in simulations of the type I showed earlier. When I asked Spencer to include, in addition, an excitatory drive representing a stimulus that ramps up over time and then ramps down, he got his simulated neuron to produce the spike trains shown here, along with the firing rate histogram shown below, much like real data. But when some of the background excitatory and inhibitory inputs were oscillating, the neuron produced the spike trains shown on the right, which have a somewhat elevated firing rate. The elevated firing rate due to the oscillation is easier to see in the plot shown here. The reason the firing rate increases is that the oscillations force many inputs to arrive roughly at the same time, effectively making them correlated rather than independent, which increases the random walk variability, and that in turn increases the slope of the drift toward threshold, making the threshold crossings occur more frequently. So, according to this theory, paying attention creates oscillations that drive synchrony and enhance the effect of the stimulus. The point is, at least it's pretty plausible that oscillations could drive synchrony and thereby facilitate communication across brain areas. And as I said, there's a lot of experimental work related to this as well. However, when we look at real neural spike trains, their inherent variability makes synchrony detection a subtle statistical problem. This figure displays spike trains from two neurons, labeled cell 1 and cell 2, recorded together across 125 repeated trials. They are part of the V1 data set collected and analyzed by Ryan Kelly, which I showed you earlier in response to the monkey viewing a movie. Here, the circles indicate the spike times when the two neurons fired within 5 milliseconds of each other. We can see the statistical subtlety of the problem immediately. While there are over 2,000 spikes in this display, there are only 35 synchronous pairs, there's no obvious pattern to them, and even if these two neurons followed independent point processes, they would sometimes fire synchronously, meaning within 5 milliseconds of each other. The question is, do these neurons fire together more often than would be predicted by chance? And the challenge is that each neuron's firing rate is always fluctuating, due in part to the changing visual stimulus and also due to other fluctuations in the neural network. But when firing rate increases, there are more spikes, and therefore there would be more opportunities for the two neurons to fire together, even if they were independent. So any reasonable statistical test for synchrony must take account of the fluctuating firing rates. Statisticians have followed two approaches, both of which assume the spike trains are generated by point processes with varying intensities, and then they apply a test of independence. One approach uses point process regression, the other resampling. I've spent considerable effort on the first approach, synchrony assessment based on point process regression, which I don't have time to talk about. The approach has generated the multiple publications listed here because the physiology and statistical modeling can be somewhat complicated. But the idea is simple. We introduce covariates to define the intensity functions, we fit the spike trains, and then we see whether there is excess synchrony beyond that predicted by these intensity functions. That is, we test for independence having assumed some family of intensities, the family of intensities being defined by the basis functions we used in the generalized regression model. The second approach to synchrony assessment applies a method called jittering. It again assesses whether synchronous spiking occurs more than would be predicted by chance in the presence of a fluctuating firing rate. It's not based on explicit regression modeling, but is instead more non-parametric in flavor, applying a kind of bootstrap hypothesis test justified by conditional inference. It was developed by Stu Gieman of Gibbs sampling fame, together with two of his graduate students, Han Amarassingham and Matt Harrison, who are now themselves tenured professors. Here, pseudo-data are created by jittering the original data, changing the spike times by some small amount, which is random. More specifically, we pick one of the two neurons and replace each spike time by a new one drawn from a distribution centered on the original spike time so that the spike time is changed by a small amount, small enough so as not to change the slowly fluctuating firing rate exhibited in the data, but big enough to destroy the existing synchrony pattern. For example, each spike time might be replaced by one drawn from a uniform distribution on an interval 20 milliseconds long, centered at the original spike time. This would tend to destroy patterns using a 5 millisecond definition of synchrony, as I showed you. Yet, because firing rates very rarely change substantially in 20 milliseconds, such jittering would change the fluctuating firing rate by only a negligible amount. It would thereby create pseudo-data from a relevant null hypothesis. The interesting thing is that in the literature on jittering, two different strategies have been proposed. The first would jitter each spike separately. The second would instead jitter spikes only within specified windows, holding fixed the number of spikes within the window. Only the second is justified by conditional inference, not the first. The second strategy is the one adopted by the statisticians. The distinction between these two strategies is illustrated here. If we have 3 spikes within a specified time window shown in red, the first strategy would allow one of those spikes to move outside the time window. The second strategy would not. The second procedure is justified, as I said, according to principles of conditional inference, in this case applied to what is called a COPSS process. In the simplest case I'm illustrating here, it would be a Poisson process with an intensity that is constant within time windows but stochastic across time windows. It is not clear what kind of point process representation could be used to justify the first procedure. In fact, a recent paper has shown that even though these two procedures seem similar, the precise form matters in the sense that the first strategy does not correspond to any reasonable point process null hypothesis. And, in addition, the first strategy can behave poorly in some circumstances, yielding tests with greatly inflated error rates. Okay, let me now recap what I've shown you and why. I've presented 2 scientific questions that have posed interesting statistical problems and have led to novel data analytic techniques. I discussed these particular examples in support of my primary message that the brain sciences are remarkably underserved by statistics. I'm now going to elaborate. Applications of the statistical paradigm in the brain sciences are rare, which is ironic in that neuroscience is full of statistical ideas. For example, to describe ion channels, Markov models are used. To understand two-choice decision-making, there's a theory according to which a particular part of the brain evaluates a likelihood ratio and another part of the brain computes the sequential probability ratio test. And a popular theory of sensory processing is based on Bayes' theorem, which led to a proposal of specific neural mechanisms that might implement Bayes' theorem. So it's ironic that in the analysis of neural data, infiltration of statistics has been so slow, yet it's explicable. I mentioned earlier that most of my colleagues, my co-authors on this review article, are by training physicists. There's lots of interesting biophysics involved with the functioning of neurons, so there's lots of physicists in neuroscience. Because these people tend to be very good at math and computation and looking at data, novel analysis methods are devised mainly by physicists. In principle, that's fine. Many physicists are statistically savvy data analysts, that is to say, they're also good statisticians. The problem is, many are not, or at least they sometimes may not recognize a need to provide a solid foundation for a method. As a result, when I first got into this subject, Emery Brown and I commiserated at great length about our shared perception that work reported in neuroscience articles, even when involving sophisticated techniques, often fails to make full and effective use of available data. Quantitative analysts who lack advanced knowledge of statistics often devise clever methods, but their whole approach to data analysis is different. No such person would ever come up with the methods I have outlined for you here. Faced with patterns of replay, they might devise some metric that measures how far the replay pattern is from the behavioral pattern. To assess synchrony, they might analyze cross-correlation, which limits their ability to build in known features of physiology. Or, as I indicated, they may determine a pseudo-data procedure that has no apparent statistical foundation and thus sometimes leads to inflated error rates. This kind of thing makes us statisticians indignant. And I like to pick on statistically naive physicists partly because they're generally so very smart and capable, but also because everyone respects physics and understands that training in physics imparts deep knowledge. Yet, somehow, when you see quantitatively savvy but statistically naive data analysts plunge ahead, oblivious to statistical principles, it seems as if it's never occurred to them that there is actual knowledge within the field of statistics. So I started asking myself, as well as every statistical colleague I could find over several years, what is different about the way those with statistical training approach data analysis? And the answer I found, which is my own way of describing what I believe is the consensus opinion in our field, is that there is, as I've been saying today, a statistical paradigm, and its most fundamental component can be summarized concisely by two core principles or tenets. First, statistical models are used to express knowledge and uncertainty about a signal in the presence of noise via inductive reasoning. Second, statistical methods may be analyzed to determine how well they are likely to perform. Now, you may quibble with the precise words I've chosen here, but I would be very surprised to find any serious disagreement with the content of what I'm trying to express. To be clear, when I speak of statistical models, I do not necessarily mean parametric models. I'm including implicitly any use of probability distributions to describe variation, along with the deceptively simple and historically highly non-obvious idea that probabilistic description of variation can lead to quantitative evaluation of knowledge and uncertainty. Also, when I talk about my perception of the neuroscience literature, while I am observing that explicit modeling assumptions are rarely made, that by itself doesn't mean the ideas people come up with are bad. They might be good. The problem is, we often don't know because no statistical formalism has been introduced to evaluate the performance of the procedure. I took the trouble to write this down because I think it's helpful to have a concise statement, and I also think we should all carry with us some kind of summary of the statistical paradigm in a form we are comfortable explaining when the opportunity arises. To repeat, my opinion is that this simple statement of the most fundamental tenets of the statistical paradigm does a pretty good job of summarizing what we statisticians bring to the table, answering the question, what is different about the way those with statistical training approach data analysis? But please recognize that while my particular phrasing comes from a contemporary perspective, the image of statistics it represents is largely due to Fisher. He began the project in his 1922 and 1925 papers and articulated it in more general terms in his hugely influential book, Statistical Methods for Research Workers, and especially in his final book, Statistical Methods and Scientific Inference. Many Fisher lecturers have discussed aspects of Fisher's concept of inference or the role of statistical models. As I reviewed the published versions of these lectures, I was struck first by the attention paid to details of Fisher's logic, and second by the reality that authors are not only people with their own perspectives, they also serve as labels on the time axis, indicating a stage of development and discourse within the discipline. Now in 2017, things have advanced to the point that we can, and on some occasions I think we should, afford ourselves the luxury of sweeping aside the details and stepping back to take a panoramic view of Fisher's work. When I do that, I find this framework summarized here to be Fisher's great legacy. I have more to say about it, but before I do, I want to touch on one additional neuroscience question. How does the reward system function differently in psychiatric disorders and addiction? This will give me a chance to leave you with a very serious statistical challenge, and it also returns me to something I said earlier, that at the present time we have no detailed understanding of brain disorders. At some point in the future, say 50 years from now, science and technology will provide vastly improved therapies. Tools that can move us in this direction have already advanced enough that the outline of how this might work is coming into focus. We can expect there to exist technologies that combine specific behavioral contexts, perhaps using virtual reality, together with targeted drugs and spatiotemporally precise disruption of neural circuits, all personalized partly through genetics. I'm going to focus on this part. The figure is supposed to show a non-invasive method known as transcranial magnetic stimulation, which is currently being used, but its effects are spatially diffuse. Instead, I'm imagining something that can target less than a cubic millimeter of tissue. And it's not a great leap to assume that in the future, not so very long from now, clever engineers will have invented some such technology. In fact, invasive methods for circuit disruption already exist. They're used mainly in non-human animal experiments. The problem is, to take full advantage of this sort of thing, we'll need greatly improved statistical methods. I want to give you some idea where the challenges are and why they're so important. To do this, I'm going to be concrete in the context of the system in the brain that assesses and transmits value, the reward system. I'm picking the reward system partly because it's heavily studied both in humans and in other animals such as rodents. In fact, human and rodent reward systems are similar in some important ways, which shouldn't be too surprising because reward drives all behavior. Everything we do is either to gain something we find rewarding or to avoid some sort of discomfort. This includes things we share with all other vertebrates, like finding food to eat, as well as things that are uniquely human, like making the commitment to sit through an hour-long lecture, where the reward is internally generated from some sense of purpose we've constructed for ourselves. In any case, another reason for picking the reward system is that being central to behavior, it's involved in many disorders of the brain. A typical picture of the way various parts of the reward system interact is shown in this figure, and it's very helpful, but it's static. There's no sense of timing here, even though behavior is constantly changing and the timing surely matters. To be effective in understanding neural circuits, with a goal of focused circuit disruption, we will need dynamic versions of such pictures. This poses a deep and challenging problem for statistics, the problem of dynamic network analysis. We want to be able to describe the evolution of neural network activity during behavior. Now, let us assume we will have recordings from multiple neurons in each of many brain regions simultaneously. This kind of thing is just beginning to happen, and it will become common in the next 10 years using a variety of technologies, including not only a variety of electrode arrays, but also techniques that image fluorescent signals emitted from labeled sets of neurons when they fire. Shown here is an endoscopic device that can be inserted deep into an animal's brain. It's basically an iPhone camera on a fiber optic cable, and it's very light so that it can be used on a behaving mouse. Optical methods are not only becoming important sources of neural data, they are also being used to either drive or shut down specific components of circuits so that causal experiments can be performed. Thus, we have a great opportunity to study networks of neurons as activity evolves during behavior, including designed circuit interventions. I'm going to be even more concrete about the problem of dynamic network analysis. Here's a video of a rat performing a typical rewarded behavioral task while a bunch of neurons are being recorded. When the light on the left lights up, he has to poke his nose some number of times until the light goes off and the one on the right comes on, and then he goes and gets his reward on the right. Now, hypothetically, suppose we were studying the rewarded behavior of an addicted animal. And suppose further that we had recordings from many different brain areas simultaneously. We want dynamic methods that, at the very least, could allow us to draw evolving pictures that, hypothetically, might look like this at the start of the trial, like this when the light cue comes on, like this when the nose poke action occurs, and like this when the reward is consumed. Then we want to change the behavior, to cure the addiction, so to speak, by disrupting the circuit at specific places and times. At the start, we might disrupt here, shown with X's. When the cue comes on, we might disrupt here. When the action occurs, we might disrupt here. And when the consumption occurs, we might disrupt here. This is not fantasy, but rather it's an extrapolation from simpler experiments that have already appeared in the literature. And I'm sure this kind of approach and data will be available soon. The problem is, we're not ready. We need tools for describing and understanding the flow of information across multiple areas evolving during a task, based on noisy data from multiple signals within each of many areas. We can and should begin with existing time series, point process, and multivariate analysis approaches, including all the very nice methods associated with Granger causality, graphical models, and network methods, and many variations of these. However, please believe me when I tell you these tools are inadequate. I don't have time to explain, but we need new ideas. All of this makes me desperate to get more statisticians involved in the brain sciences. Let me try a visualization. I readily admit that a big challenge for anyone venturing into the brain sciences is that they include many diverse disciplines, which means there's a lot of terminology and new perspectives to learn, and you also have to figure out how all these disciplines fit together into the bigger picture. But I've been at this for almost 20 years now, and I think I can help by supplying a statistician's view of the way all these disciplines make up the whole of the brain sciences. So here's my picture. And I've just finished explaining that we need help. Let me make one final comment with a few more slides. Much of what I've said today about the importance of the statistical paradigm applies throughout modern life. It is relevant to the way we communicate statistical ideas, to our teaching, and to all our interactions with others. I claimed that it's helpful to identify our shared contemporary view of our discipline. I said it had two fundamental tenets, and I identified this view as Fisher's great legacy. I also believe there's a simple big picture graphic that helps to explain the way practicing statisticians think about the role of statistical models. On the one hand, we have the real world of data. On the other hand, there's a theoretical world, a world that necessarily abstracts from the real world using mathematical formulations, which is where statistical models live, as do all scientific models. To be useful, models must abstract and simplify. This is an essential part of what we mean when we say we are building a model. Over the past few years, I've been bothered by the realization that introductory statistics courses do not emphasize this kind of big picture behind statistical practice, and I think they should. I've labeled this failure of our teaching the gap between statistics education and statistical practice. It's really important for students to appreciate the abstract theoretical nature of models and the concept that we do what we can to assess whether the models seem to be reasonable representations of the data, but we don't expect them to be perfect. This picture is supposed to remind us that they can't be perfect because models live in a completely different world, the world of mathematics, not the world of experimental measurement. Furthermore, this picture is different than the familiar population sample picture used by so many textbooks and teachers. And I would claim that the real versus theoretical world distinction does a much better job of capturing Fisher's intention. Fisher spoke of a hypothetical infinite population as in the beginning of his 1922 paper, where he said that the objectives of statistics are accomplished by constructing a hypothetical infinite population of which the actual data are regarded as a random sample. He used similar phrases in many other places. And while he often dropped the word hypothetical, I think any reasonable reading of what he wrote would conclude that he intended the modifier hypothetical to be there implicitly. In fact, Fisher railed against the subsequent description of random sampling in the sense of finite population sampling and quality control, which had entered statistics as a way of explaining the Neyman-Pearson framework. In several places, Fisher argued that general scientific inference should not be understood in terms of what that picture may suggest, namely that random samples should be considered analogous to what were called acceptance procedures in quality control, where the objects making up the population were known as lots, which would then be sampled and examined. In his 1956 book, he said, "Where acceptance procedures are appropriate, the population of lots of one or more items, which could be chosen for examination, is unequivocally defined. The source of supply has an objective, empirical reality, whereas the only populations that can be referred to in a test of significance have no objective reality, being exclusively the product of the statistician's imagination." In our contemporary context, I like the word theoretical better than hypothetical, but my intention is the same as what I understand Fisher's to have been. So let me add this picture to my interpretation of Fisher's great legacy. What I've tried to do today is tell you the big lessons I've learned applying statistics in the brain sciences, which have led me to the conclusion that brain research is underserved by statistics. I've shown you just a few examples of the way the statistical paradigm has contributed to research in the brain sciences. I've indicated it could contribute much more, and I've added here at the end my opinion that introductory statistics courses should emphasize this kind of big picture. But Fisher also realized that this perspective, which is the starting point for statistical practice, goes beyond science and beyond statistical pedagogy. This kind of statistical thinking, the internalizing of the statistical paradigm, contributes not only to science, but to citizenship. Writing in a different era and worried about totalitarian challenges to science, Fisher put it this way: "There is something rather horrifying in the ideological movement represented by the doctrine that reasoning, properly speaking, cannot be applied to empirical data to lead to inferences in the real world." He went on, "The intellectual freedom that we in the West have taken for granted is now successfully denied over a great part of the Earth's surface. The validity of the logical steps by which we can still dare to draw our own conclusions cannot therefore in these days be too clearly expounded or too strongly affirmed." Worries we now have about our contemporary social, political reality are different, but they are equally troubling, or as Fisher said, rather horrifying, and must lead us to the same commitment Fisher suggested. Let me be clear, I'm not talking about any specific political party or elected officials. I'm not. I'm speaking about our citizenry, where there is a widespread inability to appreciate or reluctance to accept that data drive progress, and wherever there are data, progress requires grappling with uncertainty. In Fisher's words, following the logical steps by which we draw conclusions. So again, what does statistical thinking contribute not only to science but to citizenship? Nothing could be more important. The thing we must do a better job of emphasizing, the point Fisher was emphasizing, is that these abstract theoretical representations, when they are able to capture important features of the real world, are exactly what we need in order to make progress. A sentiment best captured by the famous quote from George Box that represents the quintessential statistical attitude: "All models are wrong, but some are useful." Statistical thinking contributes to citizenship by encouraging humility in recognizing that variation creates uncertainty, supporting appreciation of theoretical principles for turning information into knowledge and action, and fostering pragmatism through discerning that while these principles apply to the real world imperfectly, the process of combining abstract theories with empirical data is the key to progress. Other disciplines may have their own indirect ways of encouraging humility, supporting theoretical principles, and fostering pragmatism. But statistical thinking demands it. We statisticians have a special responsibility and imperative to communicate our way of thinking when we do science, when we teach, when we talk to friends or strangers, because every day, every time we look at data, we are confronted with the problem of making progress in the face of variation. And we are the ones who begin the process with the understanding that all models are wrong, but some are useful. Thank you all for listening.